Hello, and welcome to this week's Ethical Dilemma in Science, where we're going to take a look at human brain organoids. And we're going to basically start out with kind of an overview of what these organoids are, or what we can use them for in the scientific setting, in the research setting, and more importantly, what are things that are kind of going on with them, arising to the consideration of an ethical dilemma, an ethical potential conflict. And so human brain organoids, enhancing a research model, and or, because we're potentially doing one or the other, potentially both, creating a conscious entity. So what is it that we're talking about? Again, trying to pull this from current events, so a series of news accounts that came out over the last couple months, extending a little bit further beyond that, taking a look at this. Scientists develop brain organoids with complex neural activity. That's interesting. Brain tissue grows eye-like structures that see light, potentially respond to light in a kind of stimulus response kind of interaction that would be occurring, kind of in an artificial environment. And then finally, this one from October 2021, lab-grown mini brains hint at potential treatment options for motor neuron disease and frontotemporal dementia. So this is basically an incredible new model that is available for scientists to kind of learn about the nervous system, learn about how the nervous system develops, learn about how some of the disease processes work, kind of in an artificial kind of environment, looking at this within a laboratory setting, but potentially learn about how we can interact and hopefully improve kind of a disease situation, come up with a better cure if that's what we're looking for. But the ethical dilemmas come rise, and again, recent articles, 2019 up through December 2021, taking a look at this. Lab-made mini brains produce brain waves just like those of preterm babies. So that's interesting. We're looking at cells kind of growing in a laboratory setting, but are mimicking a behavior that is similar to what we see in, premature, or not premature, but preterm babies, basically looking at babies that are born, well, I guess premature by definition, but basically born very early on and looking at their normal activity. Articles coming up in Nature, can lab-grown brains become conscious? What does it mean to be conscious? What does it mean, as these organoids, these structures become more and more complex in this artificial setting? And then the article that kind of really kind of cemented this as a possible topic for an ethical dilemma in science. Human brain cells grown in petri dish learn to play Pong faster than AI, artificial intelligence. And so basically they've been able to kind of take these cells kind of grown in a laboratory setting, grown in an artificial setting, again, use that kind of stimulus response like we talked about kind of on that previous slide with, the eye able to respond, or eye-like structure able to respond to the stimulus of light. In this case, these cells are able to respond to some type of stimulus and ultimately learn to play Pong faster than a computer programmed artificial intelligence that is attempting to learn to play the game as well. Now, ultimately, the computer is probably going to be a lot better at playing the game than a, kind of group of neurons, brain cells that are growing in a petri dish. But it does come up with that question, what are we doing in the laboratory setting that these things are becoming more and more complex, and they're starting to mimic some activities that we could, kind of associate with, kind of a human brain activity, human, potential awareness, whether we're talking about, kind of very simple brain waves or the ability to, in this case, play a, 1970s era video game. So, kind of take a step back. And so how do scientists and clinicians and doctors kind of learn about what's going on within the brain and the nervous system? So we're interested in learning about what's going on. One of the first approaches is to kind of measure the electrical activity. The activity of the brain can be measured in a series of brain waves, kind of similar to those brain waves that were generated by the preterm infant, where you basically put this electroencephalogram kind of receptor, a series of little electrodes kind of in a cap, kind of a around the position of the skull. And they're able to measure the neuronal activity, the brain cell activity, as a series of kind of electrical waves that are present within the brain. And we can look at that, and you can see, kind of awake versus relaxed versus deep sleeping. You can see different types of behavior that are going to be present within these waves. We can also go, and again, within a human patient, put a patient into some type of imaging machine and look at like a CAT scan over here on the right-hand portion of the slide. Along the bottom of the slide, we got a kind of specialized process where we can actually go through and take a look at the activity level. And so this would be like a PET scan or a functional MRI scan, where you can take a look at it. That the normal brain activity is looking at it, kind of red is going to be high activity, yellow and orange is kind of medium, and then the greens and the blues are going to be low activity. And so what you can see with this, again, using this type of imaging on a whole person, you can see kind of the normal amount of brain activity kind of measured within this imaging system. An individual that has, kind of mild cognitive impairment, so, a little bit of a disease process kind of impairing the function of the brain. And then finally over here, an individual that's suffering from Alzheimer's disease. And you can see a pretty significant decrease in the brain activity when you compare it to both mild cognitive impairment as well as the kind of normal activity that we would see within a healthy brain. So we have these mechanisms where we can study the activity within the brains of intact human individuals, but it doesn't give us the ability to, kind of really go in and kind of study it in a very specific way, be able to look at the interactions of the cells, the interactions of molecules that are involved, the ability to potentially test possible drugs for the treatment of Alzheimer's disease. We need a whole lot more information before we can go through and start providing that to a human patient, even if we're doing that within a clinical setting. So a lot of times what we're going to do is do some research on either cells that have been isolated from human tissues, maybe use an animal model in some way. But these brain organoids are kind of an interaction that allows us to go through, study human cells, the interaction of these human cells in an artificial environment, in a laboratory setting, but be able to look very specifically at how these human cells respond as opposed to animal cells. So what is it that we're doing in the laboratory setting? So what we've got here, an individual here is a donor kind of starting out, and with the purposes of this ethical dilemma in science, we're going to bypass the source of the cells. There's some ethical concerns associated with that. But ultimately, scientists are coming up with ways in which they can kind of induce normal body cells to undergo some type of specialization to become some type of cell within the body. And so these organoids, this ability to, kind of harvest cells from kind of one source, potentially a patient that needs to be treated, grow those cells up in a laboratory setting and come up with kind of a collection of cells, or ultimately a lab-formed organ that would be available to replace cells or replace organs within that patient. So we're going to bypass the source of the cells for now. But basically what's going to happen is we're going to take cells from an individual. We are going to, kind of dissect that region out. So this may be, kind of like a biopsy specimen, maybe like a skin sample or connective tissue sample. We're going to grow these cells within a laboratory setting. And so we basically separate them out. We put them into a petri dish, so a simple plastic dish here. We're going to provide them with oxygen and nutrients and some of the other, kind of basic things that they would get kind of if they were in our intact bodies. And then we're going to allow them to grow. And so for many years, what we did was take a look at two-dimensional cultures, kind of growing these cells kind of on the bottom of one of these petri dishes and looking them out, growing out, kind of along the height of it or the width of it, kind of going back and forth in two dimensions. And when we do that, we can study the different types of cells within the nervous system, and in some cases look at how these cells are going to be interacting with one another. And so the top image over here on the right-hand slide is looking at a two-dimensional culture of some nerve cells and other support cells that are going to be present in there. So it's a little layered. So it's got the support cells underneath it in the reddish, the kind of yellowish brain cells, the neurons kind of above it, and you can see they're kind of interacting with one another. They're interacting with the cells kind of below them, and we can look at this, in a very basic sense. But there are limitations. If we start looking at how these cells are working within the body, they're not in a two-dimensional structure. They're going to be in a three-dimensional structure. So they have height and width and depth associated with that, where we're not going to see that within a two-dimensional culture. And so the goal is to come up with, a model system, come up with a way in which we can look at these cells experimentally, but in a laboratory setting. And so over the last, 10, 15 years now at this point, scientists have begun, starting out with these individual cells, but growing them under specific conditions that allow them to develop into three-dimensional clusters. And these three-dimensional clusters then can grow up into these structures called organoids. And so these organoids, like this kind of section through it over here, so we got a slice through it, you can see kind of a three-dimensional architecture that is going to be present. And again, the specific colors aren't important with this, but the colors are going to represent the different types of cells and the interactions that these cells are going to be playing within this laboratory setting. So we can go through and in this kind of lab-grown artificial organ, we can, look at the development of these connections within the nerve cells. We can look at what happens if we go through and treat them with, a possible drug that could cause improvement of a disease condition or slow the delay of a disease that could be occurring. And so we got this great opportunity to study human cells in a slightly artificial but three-dimensional environment and learn a lot about them. So where does the controversy come from? This idea that these cells, as they become part of an organoid, become more and more complex and start to exhibit behaviors that are something similar to what we would see in normal human brains. And I recognize that there's a huge gap between where we're at right now and a true kind of sentient, conscious, kind of artificial brain, kind of in the Dr. Frankenstein era type concept, kind of growing in a laboratory setting. But when we take a look at it, scientists and clinicians don't have a good, hard definition for what is consciousness, and more importantly, how to measure and assess whether or not an organism or an individual is going to be conscious. And so I've got an example here from a science paper. I think this one's from about 2013, so about 8 years ago now, 7 years ago now maybe. Basically looking at, how is it that we're going to measure consciousness in the absence of kind of a stimulus response interaction. And so if you have a patient in a coma, how do you determine, if their brain is active and aware about what's going on? And scientists still are struggling with this. This is a 2018 article from the journal Nature. What is consciousness? There are still these arguments as to what it is that makes something sentient, what it is that makes something conscious, and how is it that we're actually going to measure that. So then when we give rise to that situation where we've got these brain organoids, these cells that have developed up into these three-dimensional kind of artificial structures within a laboratory setting, are starting to exhibit behaviors, very simple behaviors, maybe reflexes to start with, but potentially have the opportunity or in the future, the ability to develop into more and more complex structures. And those more and more complex structures are gradually going to become closer and closer to whatever it is that we have as a definition of consciousness. And so again, popular press articles, The Guardian over here, scientists may have crossed ethical line in growing human brains. The New Scientist from 2018, why growing human brain tissue in a dish is an ethical minefield. We've got all of these concerns that are coming up. So we need to think about this. We need to think about what it means and what kind of limitations or what type of kind of controls we need to make sure that we're doing this in an ethical way. Now, I'm not saying that these human brain organisms are going to become sentient and become the equivalent of humans, but if we take a look at how we protect our research subjects, we have special rules for interacting with human subjects, whether we're doing clinical research or we're doing survey research or behavioral research. We have to go through and we have to protect the individual and promote their health and safety. We have to minimize the risks associated with it and maximize the benefits. If we're doing a laboratory experiment with animals, we again have rules that we have to have to protect the animal, to take care of it in an appropriate way, to minimize pain and suffering, to kind of maximize the benefits that even these animals are getting in a laboratory setting, in a research setting. And so, again, looking at these human brain organoids, we're not there yet, but at a certain point, are they going to get to some level of brain activity where they need to be kind of protected in some way, where potentially, if they can respond to a stimulus, can they respond to pain? Now they maybe not be able to kind of perceive the pain or a kind of a conscious kind of understanding of the pain, but they still may have kind of a response to that. And so that stimulus and response is that consciousness, is that kind of awareness and interacting with the environment around them. So when we take a look at this, again, recognizing that this is a new area of research, but it's an area that we haven't touched on before. The science hasn't touched on before. It's introducing new ideas and new concepts on that relationship between science and ethics. And so the question for this weekly ethical dilemma in science is, how do you balance the needs? Excuse me. How do you balance the needs of a scientist who wants a more complex organoid that is going to be a better model for studying nervous system development, for studying the progression of disease, and ultimately, hopefully, for being able to better treat disease, to be able to prevent the development of disease, or at least slow the progression of the disease. So how do you balance those needs for coming up with an organoid that's more complex and better mimics the human brain, with those concerns that as it becomes more complex, as it becomes closer to the normal activity that we would see with the brain, that brain activity is becoming more and more complex and could approach at least a very basic definition of consciousness. We're not there yet, but what are the things that we need to think about? How do we balance the needs between the scientists and those concerned about the ethics involved with this situation? Now, I'm not just making all of this stuff up. There's an article here from 2018 from the British Medical Journal's Journal of Medical Ethics, Cerebral Organoids, Ethical Issues and Consciousness Assessment. A book that came out from the National Academies of Science, Engineering, and Medicine last year, 2021. Starting to look at the science and the ethics and the governance. Are there rules, in essence, that need to be in place that go along with this emerging field of human neural organoids, transplants, and chimeras, where we would ultimately be taking some of these human cells and then returning them into an animal model, injecting them into the brains of potentially a rat or a mouse or some other type of organism, to basically further study what we were able to learn in the organoids in a system that is a little bit closer to an intact and living organism. Now, as always, I'll put some additional information in the links below. So thank you. Hopefully you enjoyed this week's Ethical Dilemma in Science and come back for next week. So thank you and have a great week.